Good afternoon, I'm Ncité, and I will be presenting my PhD project, Development of a Robust Pipeline and Best Practices Recommendations for Luminex Immunoassay Data. Immunoassays can be used to detect and measure the concentration of a large number of antigens in a variety of specimens for the diagnosis of microbes, disease, or illegal substances. Recent advances in immunoassays are multiplex immunoassays that can be used to detect and measure multiple analytes simultaneously from small volumes of sample on a 96-well plate powered by Luminex XMAP technology. Analysis of the multiplex immunoassay data, referred to as the Luminex data, is complex, and current analytical approaches are highly subjective. The current challenges with the Luminex data are that they are complex to analyze. Currently, there's no standard data pre-processing approach, and the data pre-processing approach is largely dependent on the operator. The aims of my study are therefore to develop tools for multiplex immunoassay data pre-processing and to implement a standardized, robust analytical pipeline. The software and data used in the study are open source software such as Python and R programming language, and then the data is de-identified multiplex immunoassay datasets available from Stellenbosch University Immunology Research Group, as well as published data available from Public Data Repository. The ideal workflow for the data pre-processing is as follows. The Luminex data is exported by the laboratory personnel, and then the following processing steps are included in the robust standardized pipeline. The Luminex data is exported using a standardized file name, including all of the columns, and in a CSV file format. Then the data is imported into R using an in-house R function and the standardized file names, as well as experimental metadata such as the clinical data. The variable names and observations are then standardized for the Luminex data and the clinical data, followed by managing replacements and duplicates. These are cases where the sample were repeated due to a pipetting error, where the first value is removed and the second value is used for further analysis. The Luminex data is then visualized for missing values in columns and columns and rows with too much missing values will be excluded from further analysis. A common type of missing value in the Luminex data is out of range below, out of range above values. These are values that are beyond the permitted extrapolation range that are placeholders for NAs since no valid reading resulted. Shown here is an example of out of range below. The out of range below, out of range above values are then imputed using a fraction of the range, and it should be noted that imputation isn't possible if missing values in either dimension exceed a limit. A modification of Winsorized transformation is then used to shrink the outliers towards the center while retaining the order, as opposed to traditional Winsorized transformation that replaces the outliers with a single value at a truncation mean determined by trimmed mean and standard deviation. Furthermore, asymmetrically distributed data is then transformed to obtain similar distributions prior to analysis by making use of different transformation techniques such as log transformation, arcsine H transformation, square root transformation, and power transformation. And lastly, the pre-processed Luminex data is then merged with the clinical data and analyzed. To conclude, the benefits of a standardized data pre-processing approach are that it provides rapid data pre-processing, it is convenient, and most importantly, provides consistent reproducible results, which are the foundations for good science. I would like to take this opportunity to acknowledge God as well as my supervisors, the Molecular Biology and Human Genetics Division at Stellenbosch University, and my funders. Thank you.
Thank you very much, Ncité, for a very interesting talk. Wow. If anyone has any questions or comments, please post them to the chat and I will read them out. So far, the question I have is your pipeline. I might have missed this. Is there, what what programming language is it based in, or is it?
Okay, so it's run in Python from R. So a Python GUI will be used, and all the processing steps will be run in R.
Oh, okay, wonderful. So that makes it quite editable or like fixable on the fly. And Natasha is asking, is it available for others as yet?
For others?
Yeah. So is it publicly accessible?
Oh, not currently, but it will be, and it will be available on GitHub.
Oh, okay, fantastic. Really, really cool that you could build something to be used in current practice. Could you tell us a bit more about Luminex and how it would be used in a forensic situation?
So the samples are analyzed in a forensic analysis. So Luminex, you can use any type of sample and for any type of diagnosis, as long as it has an analyzer that can be that's captured by the antibodies, that is then detected by the instruments, and the fluorescence intensity is then measured, and then a standard curve is generated from which the concentration is determined to determine the measurements for the unknown samples in the sample of the unknown analytes.
And so, is there quite a backlog in getting through cases that you know of?
Yes, currently it's a manual data pre-processing approach. So that's why there's this need for an automated data pre-processing approach. And also a manual pre-processing approach tend to be less reproducible.
Absolutely, yeah, and that's everything, especially with sensitive cases like this. Thank you very much for a really interesting talk. If there are any other questions, you may pop them through into the chat. And before we move on to the next part of our event today, I'd just like to thank you, Nikita, and of course all of our second session speakers for wonderful talks. I think we've all learned a lot today. Thank you very much.